Hi, I'm Nathan, a laboratory scientist here at NorthGene.
And I'm Harry, a laboratory scientist apprentice here at NorthGene. Today we're going to show you how DNA testing is completed, following your samples from the moment they arrive in our lab, through processing and all the way through to when we send you the results.
As soon as your samples arrive at our D-side-based laboratory, we check them and confirm that all the necessary consent has been provided. Once valid consent has been confirmed, your samples are booked into our system. This involves telling the system that they have arrived and recording relevant key information such as name, date of birth and ethnicity. Your samples are assigned a unique sample identification number, or S number, so that we can accurately track which samples belong to you. Each person involved in the case is assigned their own S number. Once all S numbers are confirmed, samples are ready to be processed.
Firstly, we check the S numbers by scanning the unique sample barcode and ensuring this is the same on the samples, the consent form and on our system. Once this is checked, we move on to extracting the DNA from the sample swabs. To do this, we cut the tip off the swab and place it into the corresponding sample tube. Swab solution is then added and the tube is vortexed and heated to release the DNA into the solution. Once heated, the tubes are cooled, ready for the next stage.
While your samples cool, we create a plate map which tells us exactly where the S number will go on the 96-well plate. We transfer the DNA from your sample into the plate, which already contains the master mix. This is also witnessed to ensure complete accuracy. The DNA is then heated in a cycle. This splits the DNA into two strands and creates new copies in a process called amplification.
We transfer your amplified DNA to a genetic analyzer to undergo capillary electrophoresis. This machine reads the DNA markers by measuring how long or how short they are and counting how many times they repeat. These are known as STRs. The number of times it repeats is the number we get for each marker.
When the machine finishes running, we review the data to make sure all of the controls have passed. We then compare the DNA results for each person involved in the case. Typically, this includes the mother, potential father, and the child. Each child inherits two markers, one from the mother and one from the father. To determine paternity, we look at the number of these shared markers and the likelihood of the child inheriting these from the potential father. For paternity to be confirmed, the potential father's markers must match the child's, and probability of paternity must be equal to or greater than 99.99%. When data analysis is complete, we write the report with the concluding results. This is second checked by another member of the team before being sent to the email addresses on the consent form.
Fast, accurate results you can trust.